Good evening to all the participants gathered over here. Once again, I welcome you all for this faculty development program. And today we have session 6, second session of this day, which is going to be handled by Dr. S. Uday Kumar, the Assistant Professor, Department of Chemical Engineering, IT Tripati. And he's a highly renowned person in the tissue engineering field. You will experience the essence of his research when he talks. As I mentioned in the beginning of the FDP session, we have a composition of industry person as well as a research person. And here we have a best research person with us. So let us have his insights in the scaffold design of in tissue engineering. So let me, before start of the session, let me ask Mr. Prem Kumar sir to give a brief introduction about the today's resource person. Mr. Prem sir.
Thank you, sir. So, good evening, everybody gathered here. So, our general pleasure to welcome Dr. S. Uday Kumar, sir, Assistant Professor at IIT Tripati, to our esteemed gathering, sir. So, Dr. Kumar is a renowned expert in nanotechnology, biomedical engineering, theranostic, with a remarkable track record on innovation, research, and excellence. His pioneering work has led to multiple patents, including groundbreaking invention in biomaterials and nanomedicine. A prolific researcher, Dr. Kumar has authored 38 research articles in high-impact journal and 1,200 citation and earning an impressive H index of 19 and I-10 index of 25. This research has been showcased in journal cover pages and featured in a prominent media outlets. Today, we are honored to have Dr. Kumar Sir share his expertise and insights in scaffold design in tissue engineering. His presence is a testament to his commitment to advancing biomedical nanotechnology and translational medicine. Please join us in extending a warm welcome to Dr. S. Uday Kumar, sir. Welcome, sir.
Thank you. Thank you so much.
And Dr. Professor Uday, before the
Yeah,
start of the session, I would like to introduce our participants to you.
sure.
Here we have a combination of different discipline.
Correct.
We have participants from biotechnology, biomedical engineering, and
Mhm.
computer science engineering, and
Okay.
chemical engineering as well.
I
And
see.
we also
Great.
have participants from different region, from Tamil Nadu, Kerala, Andhra Pradesh, Uttar Pradesh also. So we
Nice.
have a very diversified participants here, and this will
Nice.
your research experience will definitely enrich the faculty community here. And
Thank you.
now your
Glad to know.
session is the session is over to use that. Thank you.
Sure, sure. Okay then. Thank you so much for the wonderful introduction. Um, can I just share my slide now or?
Yeah, yeah. Please now share. You can share this.
Okay. Yeah. Okay. I hope everybody can have a look at my screen. Is the
Yes.
presentation visible to everyone?
Yes, we could see
Awesome.
it is very clear.
Awesome. That's great.
Yeah.
That's great. Okay. Thanks for the wonderful introduction, as well as thanks for giving me an outline of the audience over here. Uh, so, good evening to everyone who are sitting across their laptop all across our nation. I would like to thank, first of all, I would like to thank the faculty members and the organizing committee for giving me this opportunity. It's, of course, a matter of privilege and pleasure to have an opportunity to interact with all of you. So in that sense, I would like to thank Dr. Aman Raj and reaching out to me and for taking me into consideration for this event. Of course, I would have been happier if I had been there in person to deliver this talk. But however, I think all of us have got accustomed to this online mode of delivering talks. So with that note, let's get started with today's lecture. So, in the next 40 minutes or so, I'll be briefly talking about tissue engineering scaffolds and some of the key aspects of scaffolds that are typically used in tissue engineering field. Okay, and basically, it's actually the scaffold design that we'll be focusing on because this scaffold design aspect determines the success of the scaffold upon implantation into the patient. Okay. And while I discuss about the design aspects of the scaffold, I'll try to point out the potential of AI and computational techniques in addressing many gaps that exist in this field of research. Okay, so now let's dive into the presentation. So moving on forward, first of all, the first thing to understand here is why we need tissue engineering, for which I would like the audience to imagine a situation. Imagine taking one of your near and dear ones to a hospital where when they are sick, and after going through a series of tests, the physician comes and tells you that their organs are failing, and it's a life-threatening situation. And then the worst part kicks in. The doctor also tells you that you might have to wait for years to get the organs, especially for the transplantation purpose. So this devastating situation happens every day throughout the world. In fact, as you could see over here in this plot, let me pull out the pointer. In fact, as I said, as you could see over here in this plot or the graph, in US alone, nearly
Professor, sorry to interrupt you.
yeah,
We could
please.
see your screen, your I mean your browser, but we cannot see your
Is it?
presentation
Oh,
screen.
okay.
Yeah.
Thanks for pointing out.
If
Okay, let me try to fix it. Just give
yeah,
me a minute.
if you could share the whole screen when you present, that would be fine. We can see everything.
I'll try that again.
Yeah.
Share entire screen. Okay, awesome. Okay, just a second. Okay, is it visible now?
Yes, yes, it is
Okay,
visible. Yeah, your slides
okay,
are visible now.
okay. Thanks
Yeah,
for
it is
letting me know.
perfect. Thank you. Yeah.
Yeah, thanks for
Okay.
letting me know. Okay, anyway, as I was just telling that before we get into the topic of the scaffold designs for tissue engineering, we just have to understand why we need tissue engineering, the field as such. For which I was just mentioning that I would like to think of a situation um, in everyday life. Yeah, pardon? Any questions or any issues with the presentation?
No, no, some, no, some side talk. Please carry on. I'll mute them.
Oh, okay,
Sorry.
okay, okay, fine. So I was just mentioning about a situation that imagine taking one of your near and dear ones to the hospital when they are sick, and then when you go to a hospital, typically you're uh, told to go through a series of tests, and after the series of tests, the results come in, and the physician comes and tells you that the organs of the patient are failing. Suppose and it's a life-threatening situation. And then the doctor also continues to explain further that you might have to wait for years to get replacement organs for the patient. So this devastating situation happens, as I said, throughout the world, almost on everyday basis. So in fact, this graph clearly outlines the same situation. As you can see, um, in in this plot, in US alone, there are nearly 110,000 people who are waiting for the organ transplant, which is indicated over here by the pink color region, whereas only 20,000 organ donors are available, which is shown over here as yellow color region. And as you see here, this gap continues to widen every year as we proceed further and further. So now I would like you all to imagine the same hospital situation in few years in future from now, when the physician comes and tells you about the organ failure of the patient, and then he does it casually without any sign of regret. And that's a bit awkward, right? Patient's organ is failing. When the doctor comes and tells you that, he has to have a bit of regret or a bit of sorrow while communicating that news. But unlike that, in future, when such situation arises, the doctor comes and informs the near and dear ones that the patient's organs are failing, but he does it without any sort of regret. That's a bit awkward, right? But I would suggest it is not so awkward because at that point of time, he would ideally have a reason or a message to give you. apart from the decision. He tells you to go home and come back in few months to fetch a fully functional organ ready for transplantation for the patient. And that is the future of tissue engineering, where we can collect cells from the patients, create complete organ in the lab for transplant, and the patient comes back to receive the organ at a later time point. The interesting thing to note over here is that we have not yet reached that stage. And you, the audience, given the fact that all of you are interested in this field, can be part of this change. In fact, I would say the change has already begun. As we all know, every beginning always needs a breakthrough. And for the field of tissue engineering, the breakthrough was given by this mouse, what you see over here. I hope most of you would have already seen this picture. In fact, a special public photograph session was organized to snap this special mouse. And it's quite obvious why it's a special mouse. You could literally see a human ear growing on the surface of the mice. So I would say this is a modern day chimera. Uh, Charles Vacanti, a researcher at the University of Massachusetts, in collaboration with MIT, made this happen. And what he did was he made a ear-like mold from biodegradable copolymer scaffold of PLA and which is polyglycolide and polylactide. And he seeded the scaffold with nearly 100 million chondrocyte cells, which were harvested from cow knee. So chondrocytes are basically the cells which are present in a typical cartilage. Your ear is basically made up of cartilage tissue. So, and the cells which it is made up of is called chondrocytes. So these chondrocytes were collected from cow knee and then implanted on a scaffold, which was designed aesthetically just like a human ear. Okay, and after seeding these cartilage cells on the scaffold, the ear was implanted subcutaneously, which means underneath the skin, in this nude mice. Okay, and after the implantation, the mice was allowed to do its usual everyday activities for nearly 4 to 5 weeks. And after 4 to 5 weeks, this ear or the scaffold that was implanted in the mice was taken out, and an histological evaluation was performed on the scaffold, which means it was evaluated whether the seeded cells were growing on the scaffold or not after nearly duration of 4 to 5 weeks post implantation. And upon histological investigation, it appeared that the scientists could clearly see the presence of mature cartilage covering the scaffold with organized chondrocyte nodules. Okay, and that showed that you could create an ear, but obviously this is not a functional ear. It is meant just for the aesthetic reasons. It appears like a ear, but it cannot enable the patient to hear the sounds and noises. Okay, so just in case a patient has lost the complete ear, you can make use of such an ear and implant it in a patient. Although it is not functionally useful, it will give an aesthetic sense and make a person look normal. Okay, so it cannot help you with the listening or anything, but anyways, it was an exciting start to the field of tissue engineering. And this was done in the year 1997, by the way. And since then, there have been many other studies along this line where people have grown organs and tissues in the lab and then implanted in a patient who desperately requires an organ to be transplanted under different situations. Okay, so I assume from the workflow of this Vacanti mouse. This mouse was basically named as Vacanti mouse. Okay, so I assume from the workflow which I described for the Vacanti mouse, one should have understood the principles and the purpose of tissue engineering. Let's define what is tissue engineering for better understanding. The pioneers defined the field of tissue engineering in a paper published in the year 1993 in the journal Science. They called it as an interdisciplinary field that applies the principles of engineering and life science for the development of biological substitutes. And these substitutes restore, maintain, or improve the tissue function. I'd like to emphasize the word interdisciplinary over here in this definition. In fact, the pioneer authors, which I'm talking about, who are part of this article in the Science Journal, stand a true testimony to the word interdisciplinary. It was written by of course, the article was written by the scientist Robert Langer and Joseph Vacanti. Joseph Vacanti is a medical doctor, and Robert Langer is a chemical engineer. So one expert from tissue processing or tissue engineering, and the other expert from the engineering materials side. Because obviously, tissue engineering involves a scaffold which requires an expert from the engineering domain and a person with a good background in life science or biology of cells. Once these such scientists come together, this interdisciplinary field of tissue engineering emerges. So altogether, these two scientists defined the field of tissue engineering in the early stages. And as I mentioned earlier, the ultimate purpose of tissue engineering is to develop or grow organs which can cover the shortage of organ donor. I don't know whether all of you had a chance to look at the first slide, which I mentioned, where I mentioned that there is a clear organ shortage situation across the world. The number of donors are much lower than the number of patients who require new organs. Okay, and this gap can be covered by the field of tissue engineering. Okay, so the ultimate purpose of tissue engineering is to develop lab-grown organs which can cover the shortage in organ donors for transplantation procedures. And the other interesting advantage of this strategy would be that it would not require any immunosuppressive therapy. I would explain why it is so in the following slides a bit later. So some of the other interesting applications of tissue engineering would be to generate in vitro models equivalent to that of each organ, which can then eliminate the need for animal testing and trials. For instance, if there are a lot of studies going across the world where people study diabetes, people study liver disease and all those things. And for all these studies, we make use of mice or any other kind of animal models. So if we are able to design a scaffold which can mimic the in vivo conditions, the conditions in which our heart cells are actually in within our body. If such conditions can be mimicked in a lab scale, we can grow the heart cells in the lab on such a scaffold and mimic the conditions in which it is actually present within our body. So once we are able to mimic the conditions that are present in our body in the lab level model, then I think we can do all these disease-based studies in the lab scale itself instead of making use of animal models. So that is also another potential application of tissue engineering. We can eliminate all the animal studies if we are able to exactly mimic the conditions that exist in our body and then develop organs in the lab and use these organs for testing different drugs, let's say for against diabetes or liver degeneration and all those things. Okay, so some of the so the in vitro models can act as an equivalent to each organ, which can then eliminate the need for any other additional animal testing or trials. So this drastically can reduce the cost and time for pharmaceutical companies and ultimately reduce the market price of the drugs. Okay, because it's actually the clinical trials that takes the longer duration and for which the pharmaceutical industries pump in a lot of money to do these clinical trials. And once these clinical trials are done, then the drug reaches the market. So this whole sequence of events that is there as a part of clinical trial can be cut down by making use of engineered scaffolds, engineered tissues in the lab. Okay, so the other interesting application which I would like to point out is, which is very interesting, so I'd like to emphasize over here. It's a case of a Petri plate grown edible meat. I don't know how many of you heard about this, but the cartoon over here actually represents the concept which I'm going to explain over here. It's called the concept of from pig to plate, where you don't have to kill the animals, just collect the cells from these animals, grow them in the lab for large-scale meat production, and make use of it for as an edible product in the market. So you don't have to basically kill the whole pig to get the pork. Okay, so you can collect some cells from the pig, grow the cells in the lab, create a whole cluster of cells which will look like a tissue, and then this tissue mimics the exact same meat which we get from the pigs, and that can be used as an edible material. Okay, in fact, I think many of you would have heard about artificial eggs, these and which are already there in the market, in the commercial level. You can get these artificial eggs. These artificial eggs are a pure vegetarian product. They're called artificial eggs. But they are pure vegetarian. They're made from lots of different kinds of seeds and all those things, grams basically, and they mimic the exact texture and taste of a typical egg. Okay, so there are a lot of artificial products coming into the market, and one of such product is called this artificial pork. Okay, so That is again one of the possible applications of this field of tissue engineering. Okay, so what we're looking at over here in this slide is basically an overview of tissue engineering. So basically, when a patient with a damaged tissue comes to a doctor, what a doctor typically does is that he basically takes the cells out of the person, and then he cultures these cells so that you get enough number of these cells, and then he sees the cells on the scaffold and gives them sufficient required signals like growth factors and cytokines for the cells that are seated on the scaffold. And with all of these things combined, the doctor at the end uh, of the time duration should have a fully functional tissue which should replace the damaged tissue of the patient. And once the patient damaged tissue is replaced with this lab grown tissue, the person takes a ride home for recovery. So we try to do exactly what is there in this image in the case of tissue engineering. It sounds simple, but the challenges are like identifying what type of cells we should one choose to cultivate a typical organ in the lab, and how to isolate the cells from the patients, how to culture the cells in the lab. These patients, whatever cells has been collected from the patients, they have to be grown in the lab, right? So we have to you have to know what is the composition of the medium that is required to grow these cells in the lab, and how to design the scaffold. After growing the cells, these cells has to be seeded on the scaffold. In the case of Vacanti mouse, we saw that a human ears was the scaffold. Okay, it was made up of a polymer, PLA and PGA, and on this polymer, the chondrocytes were seeded, right? So just like that, the scaffold has to be designed, and you have to know how to design the scaffold, okay, and how to make sure the scaffolds do not get rejected. That is again another new issue. And what kind of signal should be provided to the cells so that it is able to grow just like the way it grows in a typical uh, human system, or in otherwise it can be any other animal system also. Okay, and apart from that, we also have to look at what are the biophysical cues that are required to simulate the cells so that they mimic the in vivo conditions. And with all these factors taken care perfectly, hopefully at the end of the whole sequence of events, we would achieve the desired outcome of generating a tissue ready for implantation. So no doubt the challenges are enormous in the field of tissue engineering in realizing its actual application in a real life scenario. Okay, so now that we have looked at the basic introduction of the tissue engineering, we can easily identify three basic tools that are required for tissue engineering, which are basically the cells. And then the other component which we require for tissue engineering is actually the scaffold. And the third component which we require for tissue engineering is actually the cell signalings. Okay, so in fact, these three factors are together are often referred to as the triad of tissue engineering. So if you are given a task to engineer a specific tissue in a lab, the first question would be to identify how we take these three arms of the triads. The first thing is cells, which is actually the living part of the tissue. The next one is scaffolds. Here we have very critical role. Scaffolds have a very critical role in tissue engineering, especially because they can actually direct the growth of cells which are seated on them. We basically seed the cells on the scaffolds and use these cell-seeded scaffolds as a tissue engineering construct. So mammalian cells, as we all know, are anchorage dependent, which means they require surface to adhere, and only when they adhere, they grow. So these scaffolds should enable the mammalian cells to adhere onto them or latch onto them, and then only they can proliferate to form a functional tissue. So, so basically, the scaffold or the substrate is meant it should be designed such that it can hold or adhere the cells on top of it and allow the cells to grow on top of it. Okay, and scaffold are meant for that purpose, basically to attach the cells and let them proliferate on the surface. Okay, so and indirectly we can also say that the scaffold basically provides the matrices for cell adhesion, proliferation, and as well as migration. The cells, once they proliferate, they also have to migrate and penetrate into different portions of the scaffold. So migration also is an important aspect to be taken into consideration. So ultimately, the scaffold basically provides a structural support and shape to construct the tissues or organs. The scaffold you should be basically, and another important aspect which we have to take into consideration when designing a scaffold is that the scaffold should be biodegradable as well as biocompatible. Biocompatible means it should be compatible to the biological system, and biodegradable means if it should be capable of degrading itself within the system without leading to any toxic byproducts. Okay, it should not elicit any sort of immune response, or it should not release any toxic byproducts or metabolites when it is implanted inside the body. So, such should be the nature of the scaffold. It should be biocompatible as well as biodegradable, which are typically characteristic feature of any successful scaffold. So, finally, the third one is cell signaling. This may be a growth factor. Um, Growth factor or a hormone which tells the cell when to proliferate or differentiate and how to differentiate into other type of cells. So in this lecture I would basically restrict my discussion uh, to scaffolds alone, and we'll specifically look at the key aspects of the scaffold that determine the success of the tissue engineering. So amongst these three aspects, we'll be focusing only on scaffold, and we will be looking at some of the key aspects involved in designing a scaffold, and we'll also try to understand how these key aspects govern the success of a scaffold in the field of tissue engineering. Okay. So before we understand these scaffolds and fabrication, it is also essential to understand what we are trying to mimic over here. When we design a scaffold, we have to know what we are trying to mimic over here in the scaffold. Okay, so the simple answer to this question is that we try to mimic in vivo extracellular matrix-like features. So in our body, we have cells, and the cells are growing on a matrix, and this matrix is called extracellular matrix. So when you design a scaffold, you are basically trying to mimic this extracellular matrix, so that the cells seated on them also mimic the same situation as they are present within our body. Okay, so and it's a pretty important factor that determines the success of any transplantation process. So it is essential to understand what we are trying to replicate here. And as I already mentioned, it is actually the extracellular matrix composition that we are trying to replicate in the scaffolds. So, and this extracellular matrix is what is abbreviated over here as ECM. So ECM composition, uh, in order to mimic the ECM, we have to understand what is the composition of ECM. So getting down to ECM composition can be broken down to two major classes, of which includes proteins and proteoglycans. So amongst the proteins, there are actually just two things. One is actually a structural protein, which provides a support for the tissue to grow. And amongst the structural proteins, the common ones are called as collagen, elastin, fibrillins. And the other kind of proteins are called specialized proteins called fibronectin, laminins, which act as a which have special biological functions also. Now, the other second class of extracellular matrix composition is proteoglycans, as I just mentioned a few minutes back. So these proteoglycans, they are basically molecules where you have a protein core to which long glycosyl aminoglycans or sugar molecules are attached. So these are complexes that have higher molecular weight components, and they are present typically in almost all kind of tissues, extracellular matrix. So they provide basically the viscosity and the fluid-like nature of the extracellular matrix itself. Okay, so now that we have understood the composition nature of the extracellular matrix, we should have an idea what we are trying to fabricate when we design a scaffold. Okay, and basically we have to have a scaffold that is analogous to the extracellular matrix, which we have just covered now. Okay, so different scaffold fabrication approaches have already emerged in the past, and especially to address the issues that are faced in typical tissue engineering field. And some of the recent tools used for fabrication of such three-dimensional scaffolds are the use of 3D printers. I think many of you would have used this in your research work. And these 3D printers are used for in vitro transplant implant construction, especially for tissue repair purposes. And again, many of you would have also worked with nanofibers mat for tissue engineering. Hydrogels is again another kind of scaffold which is emerging very recently. And then we also have a category called biomimetic self-assembling peptides or DNA aptamer scaffolds and so forth. So amongst all these, let's focus on the type of scaffold rather than the fabrication technique. So we'll not be focusing on different techniques that is used for fabrication of scaffold, but we'll be focusing on the different types of scaffold that can be used in tissue engineering in the further slides. So as shown in this figure, over the last 2 decades, 4 major scaffolding approaches for tissue engineering have evolved. It would be informative to highlight the working principles and characteristics of these approaches before discussing the tissue-specific consideration. So let's look at the first approach, which is called pre-made porous scaffold. This is indicated over here. The raw material which is typically used in this procedure would basically include a synthetic as well as natural biomaterial. And apart from these 2 components, there's additional component present in this technique, which is called a porogen. So porogens are incorporated to prepare this solid freestanding scaffolds. So porogens, basically, as the name indicates, are basically template materials used to create porous structures which can be easily removed after polymer solution solidified to form the complete scaffold. So once the porogens are removed, they leave a stable porous structure in the scaffold. And since the scaffold now has porous structure, the cells which you add on top of the scaffold then can penetrate into the deeper portions of the scaffold. And as the scaffold degrades, it will be completely taken over by the cells. And the cells will divide, proliferate, and migrate, governed, and all these 3 processes will be governed by the structure of the scaffold. Okay, the way you design the scaffold. So the advantage of having such a scaffold would be that we would have, we obviously would have diverse material choices, and we can also precisely design the microstructure of these scaffolds. But looking at the other side, the disadvantage of this approach would be that it is a bit time-consuming cell seeding procedure. You have to seed the cells uniformly throughout the scaffold. So when you try to do it uniformly, it becomes a time-consuming procedure. Because inhomogeneous distribution of cells should be avoided. But such scaffolds find extensive applications in both soft and hard tissues in our real life scenarios also. So intuitively, coming to the next technique for fabrication of scaffold, or the next kind of scaffolds which is typically used in tissue engineering. Intuitively, when you, as I mentioned earlier, the best scaffold for a tissue engineering field should be the ECM of the target tissue in its native state. So what we are trying to mimic is extracellular matrix in tissue engineering when you're designing a scaffold. So instead of mimicking extracellular matrix, the extracellular matrix can itself act as a scaffold for growing a tissue in the lab, right? You don't have to make a polymer-based scaffold to grow the tissue in the lab. You can collect the extracellular matrix and make use of it as a template to grow the cells in the lab. Okay, and that is what is all about this decellularized extracellular matrix technique. So in this approach, what we do is we start with the organ itself. Whatever, let's say the heart is damaged, okay, in a patient, and we want to have a functional heart to be developed in the lab. So the first thing to do over here would be to harvest the cells from the patient, heart cells from the patient. It's very small quantity so that it doesn't affect the function of the heart. And then we remove the cells from the organ by different decellularization. And on the other hand, what we can do is we can collect an heart which is not fit for donation purposes, and then remove all the cells uh, from that heart, especially which is recovered from a cadaver. And once the cells are removed, what you have left over there is the extracellular matrix. You have the exact same extracellular matrix that makes up the heart cells now. And then you have taken some heart cells from the patient who's suffering from the disease. And these cells can be seeded into this decellularized heart. And then the whole thing can grow into a functional heart in the lab. Once it grows into a functional heart, it can be implanted in the patient. It sounds like a bit of a movie sort of thing, but it has been performed in the lab. Such hearts has been grown in the lab and have been implanted in patients in few cases. We'll discuss about those cases specifically in the future slides. Okay, so basically what we do over here is we remove the cells from the organs by different decellularization techniques, but at the same time we preserve the extracellular matrix composition from the cadaver heart. Okay, and then we introduce the host patient cells in these decellularized heart, and then the cells which have been introduced, they proliferate and differentiate to form the complete functional heart. Okay, and the perfect analogy for this situation would be when you move from one home to a new home. assuming yourself as cells and the home as scaffold in the scenario. So the first thing to ensure that when you're moving from one home to the other home is that the new home should be free for you to move, right? So that is to ensure the residence has vacated. And analogous to this situation, what we do in this procedure is that here we see that the cells have been removed from the organs, or the organ has been decellularized and the cells have been removed, so that only the extracellular matrix is remaining. And then the next step when you're doing a moving process is that you move into a new home and settle down and arrange your things. So just like that, the patient cells which we collected, right? So these patient cells are moved into the new scaffold. They are allowed to settle down on the scaffold, attach onto them, and then they proliferate. And over here, the advantage of this technique over here is that uh, when we make use of such a scaffold from another living system, they mimic the composition and the mechanical properties of the tissue to the best extent possible, because there's not much artificial component over here. Okay, you are taking another useless heart which cannot be transplanted, removing all the cells from that heart, and then what is left over there is the scaffold. Okay, and in that scaffold you are implanting or seeding the heart from the patient who requires a replacement or transplantation to be done. Okay, so once the cells start to proliferate, penetrate into the scaffold, and develop a functional heart in the lab. Okay, but the disadvantage of this procedure is that again, there's a possibility of heterogeneous distribution of cells. When you take out a decellularized heart, it has complicated, intricate structures. And when you're seeding cells on this decellularized heart, you have to ensure that cells distribute uniformly throughout the heart. And because it has complicated, intricate structures, it is very difficult to ensure that the cells are uniformly or homogeneously seated on this heart. Okay, so those are the drawbacks of this technique. So now moving on to the third technique, which is called cell sheath engineering. Okay, so basically it sounds like a process rather than a technique. So let's see how it is done. So in this approach, what is done, the cells are cultured to a state of confluency, and then these cells are induced to secrete their own extracellular matrix. And these secreted extracellular matrix is then harvested without the use of any enzymatic methods. Because when you make a specific thing to note over here is that we do not make use of enzymes. That is, for instance, the protease enzymes like trypsin, which can digest the extracellular matrix. So, which we don't want to happen. We want to keep the extracellular matrix intact. So this specific technique, we have to harvest the extracellular matrix without making use of any proteolytic enzymes. So how do we do? How do we detach the cells from the extracellular matrix if you don't make use of enzymes? So for this, the cells are actually controlled on a thermoresponsive polymer. Like, for instance, one of the examples of thermoresponsive polymer is polyisopropyl acrylamide, which changes its property based on the temperature. Okay, that is why it is called thermoresponsive polymer. So once the cells are seated on such a polymer and the cells reach the state of confluence, uh, the cells are detached from this scaffold by thermally regulating the hydrophobicity of the polymer coatings. That is, this thermoresponsive polymer, its hydrophobicity can be modulated by just altering the temperature of the polymer. Okay, so once the cells are seated on the polymer, they grow to the maximum extent possible. And once they grow to the maximum extent possible, you're not interested in cells. What you're interested in is the extracellular matrix that is produced by the cells. So you have to remove the cells. To remove the cells, what is done is usually the temperature of the scaffold is raised, which makes the scaffold hydrophobic in nature. And because of that, the cells are detached from the extracellular matrix. And what we are left over with at the end is the extracellular matrix on the polymer surface. And this extracellular matrix can act as a scaffold for growing our interest of cells. Okay, and this process basically repeated to laminate multiple single cell layers to form a thicker matrix. So although the ECM extracellular matrix sheets offer a biocompatible scaffold with better cell integration, they are a bit time-consuming process, and as you can see, it involves multiple steps or multiple procedures finally to arrive at your scaffold of interest. Okay, so moving on further with the fourth strategy or the final strategy, which I wanted to emphasize over here is the fourth strategy is called the cell encapsulated hydrogel matrix strategy. Okay, so what we do over here is a synthetic or natural biomaterial is used, which is capable of self-assembling into hydrogels. Usually, gelation of such material is self-assembly process, which is initiated by a process such as or a stimuli, which is initiated by a stimulus, for instance, such as pH change or temperature change. Okay, we have many of you would have worked with hydrogels and all those things, pH sensitive hydrogels, temperature sensitive hydrogel, which change their mechanical property when they are subjected to pH or temperature changes. A very common example of one such hydrogel is one such material, which is found more commonly in the field of tissue engineering, is called matrigel. It is also used for implantation of tumors in mice, this matrigel. This matrigel has a property that when it is incubated at 37 degrees Celsius or it is kept at 37 degrees Celsius, the matrigel proteins polymerize, or basically the proteins solidify, producing a gel sort of composed material. And the composition of this matrigel also mimics the extracellular matrix proteins. Okay, so apart from this, there are a lot of other naturally occurring polysaccharides derived from algae. For instance, sodium alginate is a most commonly used material, which can again go salt to gel transformation based on the temperature. The advantage of now, when we look at the advantage of this technique, we can clearly understand that it would be we can inject such hydrogels as a one-step procedure. We don't have to operate the patient and then implant the organ. We can, since they are present initially in the liquid state, you can inject these hydrogels into the patient, and once they enter into the patient, they are exposed to 37 degrees Celsius, at which they solidify from liquid state to solid state, and then they act as a scaffold for the cells to grow. So the method of implantation is easier over here. Um, Now, but you look at, there are, of course, there are some disadvantages associated with this technique. They cannot be made to take up specific shapes of the target tissue. For instance, if you're trying to make use of hydrogel for designing an ear scaffold, they cannot be made to take up the shape of an ear as easily as compared to any other polymer or other approaches which we have discussed. Okay, so with that, let's now move on to see some of the wonders of these scaffolds. How these scaffolds go on to generate the complete organ in the lab. First of all, is that possible? Um, yes, it is certainly possible. In fact, all of us are actually living evidence of what a single cell can transform into. All of us have grown as a complete organism from a single cell called zygote, right? So, in fact, just like that, we can collect cells from patients, grow these cells in the lab, make the complete organ. Okay, so in fact, as you can see over here, a whole range of human organs have been developed in the lab with scaffolding approaches that I just mentioned earlier. And the interesting part is that many of these organs have finally found a place in the patient's body. It's not just, it's an experiment that is performed in the lab. These organs have been grown in the lab and then implanted in the patient, and they have saved life of the patients. So which means they are clinically translatable approaches. Okay, so they are implanted. So as I said, they are actually, all these organs are actually implanted in human patients. And what you see over here is basically an umbilical venous endothelium HUVEC cells. I'll not go into the abbreviations over there. Over here, they are basically HUVEC cells. They are seated in decellularized rat kidneys. Okay, what you're looking over here is actually a rat kidney. The cells that are present in the rat kidney are removed, which is what is called decellularization process. And what is left over there is the extracellular matrix. In this extracellular matrix, HUVEC cells, which are usually present in our kidney, are implanted there, and then uh, uh, and then it grows further to develop as a functional kidney. Okay, of course, you require multiple kinds of cells. Kidney is just not made up of a single kind of cells. There are a lot of other kind of cells which are required to form a functional kidney. Okay, but we are just talking about implantation of just the HUVEC cells, which is one of the cells required to form a functional kidney. And once you see these HUVEC cells, they proliferate throughout the decellularized matrix and then take the shape of a kidney. Okay, now what you're looking at over here. Is this is a tissue engineered heart wall developed using human marrow stromal cells, and these human marrow stromal cells are seeded into polyglycolic acid scaffold matrices. Polyglycolic acid is a very common polymer that is used in tissue engineering. One of the reasons for that is it is biocompatible as well as biodegradable. Okay, and this is one of the examples. Okay, where you have made a polyglycolic acid scaffold, and then the human bone marrow cells are seeded on the scaffold, allowed to grow or differentiate into specific kind of tissues that helps us in finally ending up with a functional heart wall. Okay, and what you're looking at over here is actually that the engineered bone have been developed with donor bone marrow stem cells seeded in microporous synthetic calcium phosphate minerals. So hydroxyapatite is again one of the most common material which is again used in bone tissue engineering. Again, one of the major component of hydroxyapatite is calcium phosphate. So such kind of scaffolds can be used for bone tissue engineering. Okay, now that let's dive into details of some of these organs and try to understand what it takes to develop such a clinically transplantable or translatable organ in the lab. Okay, we'll first talk about the one of the most complicated organ that has been developed in the lab, a functional heart that was developed in the lab. This was done by a group of researchers from Harvard Medical School, um, who reported for the first time the lab engineered functional heart in the year 2016. Okay, so what they did was 73 human hearts were used for this study, for performing this whole study. And all these human hearts were non-transplantable, which means these are hearts collected from the donor, but they were not fit enough to be used for transplantation purpose. So all these hearts were collected and then made use for this study. And they are called non-transplantable human hearts. Okay, so, and these non-transplanted human hearts were basically used to generate the decellularized scaffold. They took all these non-transplantable heart and they removed all the cells. And what is left over there is extracellular matrix, basically a scaffold. Okay, so deceleration of heart was basically was achieved by perfusion of different chemical reagents, and one of the common reagents which is used is SDS and Triton X. Both Triton X 100 and SDS are detergents, which basically denature the protein. But there's a difference between both of them in terms of denaturation of protein. The Triton X100 disrupts the DNA protein or lipid or lipid protein interactions. And while it does that, it also maintains the native protein structure that is present over there. Okay. Whereas on the other hand, SDS is an ionic detergent which can basically completely solubilize the cell and nucleic membrane and fully denature the protein. So basically, they both SDS and Triton X100 enable an efficient decellularization of the heart. In fact, I suggest you can perform this procedure in your lab also with the organs collected from the meat shop uh, and use that as a starting material for your research after decellularization step. You can make use of these reagents. Suppose, let's say, you get goat liver, and then you can remove all the cells that are present in this goat liver, and what you'll be left with is a non-living thing. It's just the extracellular matrix. And then you can seed the human cells. If you're growing the human cells in the lab, you can seed the human cells in the scaffold, especially if you see the liver cells, and then you can see what kind of functions, or does it proliferate throughout the scaffold, penetrate throughout the scaffold, and is it able to mimic the mechanical properties of a typical human liver and all those things. So such studies can be performed even at the lab scale, because what you basically require is an organ, decellularize the organ, and then see the desired cells in this decellularized organ. Okay, so after decellularization, for to do the decellularization, decellularization of the heart, what is done is basically perfusion. Now the perfusion decellularization heart is perfusion of these two agents, that is SDS and Triton X100, is done through the heart, such that as these two compounds travel through the chambers of the heart, they interact with the cells and the extracellular matrix, detach the cells from the extracellular matrix, and remove the cells from the heart. Okay, so now the perfusion decellularized heart is basically composed of high extent of insoluble collagen, which is basically the extracellular matrix protein, and to moderate extent of sulfated glycosylamine glycans, which I mentioned as major component of extracellular matrix, right? And it is also made up of lower concentration of alpha elastins. And all these proteins basically are, as I mentioned earlier, are part of extracellular matrix. And basically what we have at this point is a scaffold with exact architecture of an heart. Now let's see what cells were used in this study. Okay, now we have an architecture of the heart. And now let's understand what kind of cells were used in this study to make a functional heart in the lab. Okay, so the special kind of cells called induced pluripotent stem cells were used in the study. And these cells were generated from adult skin cells by de-differentiation by supplementing additional factors, growth factors, and all those things. So these induced pluripotent cells were then differentiated into cardiac muscle cells. Our heart is made up of cardiac cells, especially muscle cells. So these stem cells were differentiated into cardiac muscle cells in the lab, or cardiomyocytes. And once these cells differentiated into cardiomyocytes, they were implanted into the cells. Okay, and this was done with the cells. And let's understand what signals were used in this procedure. Okay, I already mentioned about the cardiac differentiation while describing the cells in the earlier slide. So apart from the cells and scaffold, you also need cellular signaling materials like growth factors and all those things to perform the tissue engineering aspect. Okay, so certainly the procedure involved conditioned media with different differentiation factors. So in this case, it was achieved by combining by combination of pathway inhibitors and regulators, especially the Wnt beta-catenin pathway. And with the help of the regulators of this pathway, the cardiac protein expression in differential cardiomyocytes was confirmed by doing multiple procedures like immunostaining for markers proteins like alpha-actinin. What you're looking at over here is immunostaining for the typical proteins that are found in heart muscles, heart. And what you're looking at over here is alpha-actinin and cardiac troponin T expression levels after these cardiomyocytes were developed from stem cells. Okay, so the differentiated cardiomyocytes were seated in the whole decellularized heart, and around 500 million cells were seated in this decellularized heart, and which led to the growth of proliferation of these cells on the surface of this decellularized heart. And now, instead of calling it decellularized heart, we refer to this heart as recellularized human heart. And these heart were mounted in human heart bioreactor to provide mechanical stress, which is also called as antigrade perfusion and matrix strain stimulation. So that is what we'll be able to see in this image. I don't know whether okay. So as you can see in this video, this is the functional heart that was developed in the lab. You can clearly see that um, it is subjected to mechanical stress so that the cardiomyocytes experience the same condition as they experience within our body. And when they experience a similar condition, they secrete extracellular matrix in a similar fashion as they do within our body. And then ultimately, they will finally develop into a functional heart. Okay, so this video basically shows the effect of matrix stimulation. And what we can see over here is the electrical stimulation of the heart muscles. You can see that when these heart muscle cells that were grown in the lab, when they are subjected to electrical stimuli, they contract and expand. Okay, and this electrical stimuli was delivered by making use of cardiac leads that were placed in the anterior wall of the left ventricle. Okay, so basically this ultimately at the end of the study, you could clearly see that a simulated myocardium was generated, and you could clearly see that the visible contractions in this video also. And after a culture period of, let's say, 14 days, the whole organ was repopulated with myocardial segments, and they were metabolically active also, according to the other assays that were performed in the study. And obviously, as you can see over here in this video also, they could also have the contractile function that is typically present in a heart muscle. And they also respond to electrical stimulation, as we could see in this video. Okay, so myocardial tissue regeneration was demonstrated in this study. And we can clearly see that the decellularized heart can be seeded with cardiomyocytes derived from stem cells. And these cardiomyocytes can proliferate, can attach, proliferate, and migrate throughout the decellularized heart and enable us to grow a functional heart. Functional heart in the sense that this heart can perform mechanical contraction and expansions, as well as they also respond to the external electrical stimulus. Okay, so now that we have understood the case of heart, now let's move on to another example like this. So what you're looking at over here is about a bladder. So this study was published actually in Lancet in the year 2006. In this study, 7 patients with end-stage bladder diseases were grafted with autologous engineered bladder tissues for reconstruction purposes. So the first step is to identify a scaffold that is suitable for bladder tissue engineering. So unlike the earlier procedure, they didn't make use of the bladder as such and decellularize it. Instead of that, uh, instead of that, they chose two different scaffolds. Uh, a biodegradable bladder-shaped collagen matrix derived from decellularized bladder submucosa was used, and a biodegradable composite scaffold that was made of a polymer that was made of collagen and PGA polymer was used. So two different scaffolds were used. One, a decellularized bladder. Other one was basically collagen and polyglycolic acid. Mixture of these two was used to form a scaffold for bladder tissue regeneration. Now we see that collagen one of the reasons why collagen is used in designing the scaffold is that collagen is a common component of any extracellular matrix. And collagen is a major structural protein that is present in extracellular matrix in various connective tissues that is present in our body. And it forms the major component of connective tissue, making up nearly around 25 to 30%, 35% of the whole body protein content. So I would say this is an important study from the scaffold point of view, as in this study, as they present a straightforward comparison of performance of a decellularized scaffold and a synthetic scaffold that is made up of collagen and PGA. Okay, so once both scaffolds were ready in the study, the second step would be to harvest the cells and process and expand these cells. So a bladder biopsy was obtained from the patient who is suffering from bladder issues, right? So some bladder biopsy, which means they collected the bladder tissue from the patient, and the urothelial muscle cells were separated from this tissue and grown in the cell culture dishes by making use of growth medium, just like DMEM with FBS and all those things. Okay, and the third step after harvesting the cells, that which was the second step, of moving on forward, the third step would be to seed these cells on the scaffold. So the exterior surface of the scaffold was seeded with the smooth muscle cells at a concentration of nearly 50 million cells per centimeter cube of the scaffold. Okay, so the cells that were seeded on the scaffold were allowed to settle, and then aggregate allowed to settle, attach and proliferate in an incubator for a duration of 1 hour. And then the growth medium was supplemented to this scaffold to cover the entire scaffolds in the growth medium. After 48 hours, the urothelial cells that were seated on the inside of the scaffold, um, after 48 hours, the urothelial cells, which is another kind of cells that is present in your bladder, were seated in the inside of the scaffold at a concentration of around 50 million cells per centimeter cube. The cell-seeded scaffold were then placed in a sealed container filled with both DMEM and keratinocyte growth media. Both of these media were mixed in the ratio 1:1 and grown at the typical condition of 37 degrees C in a CO2 incubator. And this was done for a duration of around 3 to 4 days. Okay, after this, after the growth period of 3 to 4 days, a small tissue sample was collected from the edge of the scaffold and examined under the phase contrast microscopy, and some of the other staining procedures were also performed. For instance, what you're looking at over here is trypan blue analysis of both the scaffolds. So trypan blue analysis was performed for both the scaffolds before actually transplanting them into the patients. And as you can see, that trypan blue analysis clearly confirmed the attachment of cells in these samples. Now, if you look at the attachment of cells in these scaffolds, as well as the proliferation of these cells in both the polymers, the collagen as well as PGA. Okay, now if you look at this promising outcome of the study, we can see that the engineered bladder tissue clearly shows the tri-layered structure consisting of lumen that is lined with the urothelial cells, and which is also surrounded by submucosa indicated by S, and the muscle cells, which is indicated by M. And then regarding the functional ability of the bladder, it was determined that the bladder leak point pressure in terms of volume and compliance increased greatest in the case of composite engineered bladders. So an outcome of this study is that in terms of mechanical property, the scaffold made up of collagen and PGA was performing better as compared to the case of decellularized bladder. Okay, so that is why this study is a bit crucial study in the field of tissue engineering, where you do a direct comparison between a decellularized scaffold and a synthetic scaffold. And at the end of the study, it was shown that if you look at the functional property of a typical bladder, the bladder that was developed by making using collagen and PGA performed better as compared to a decellularized bladder scaffold. Okay, so now we'll move on further to a very interesting case. And in this case, this case is specifically about tissue engineering of a trachea in the lab. And this study was performed by surgeon Dr. Paolo at Karolinska University in Sweden. They performed what they did was basically they performed a chest CT scan of a patient who has a faulty trachea, and they did this by making use of three-dimensional volume rendering of the CT scans, and they made use of these multiple CT scans to generate a 3D model of patient trachea. Okay, and now um, basically now if you look at the CT images, they are two-dimensional images, and making use of the sequential two-dimensional images, a complete 3D model of the patient trachea was developed. And only when you have the model of patient trachea, you can design a scaffold that mimics the patient trachea, right? So now this is where I guess AI and computational techniques can play a vital role. For instance, making a three-dimensional model of patient trachea is pretty complicated. And based on the imaging technique outputs, like for instance, CT images, we have to render and reconstruct a three-dimensional model of the trachea by making use of 2D images from the CT scans. Okay, so which obviously requires a lot of computational techniques so as to make it exactly generate a model that mimics the structural feature of the trachea of the patient. Okay, for instance, so basically they did such kind of things, and they ended up making use of this 3D model to manufacture a synthetic mold or synthetic scaffold that is made up of a glass mandrel in the shape of a trachea. Once you get shape of the trachea, making use of CT images, once they have the shape, they made a scaffold that is made up of glass mandrel in the shape of trachea. And then on top of this mold, a porous layer of copolymer called polyurethane and silicoxane was processed by making use of extrusion phase inversion method. So this extrusion phase inversion method is basically a simple one-step dip coating procedure where the scaffold is taken and dipped inside the polymer solution and then taken out. And once you take it out, the scaffold is covered with a thin layer of polymer solution. And that's polymer, basically the polyurethane and silicioxane, acts as a scaffold for the growth of the cells. Okay, so now with the scaffold ready in hand, the bone marrow stem cells were then harvested from the patients and cultured in vitro with by supplementing growth factors like transforming growth factors, granulocyte, colony simulating factors, and erythropoietin and all. Okay, these are all growth factors which help us in growing these cells in the artificial lab situations. Okay, so these growth factors were meant to basically induce proliferation, differentiation, and mobilization of the stem cells. And the differentiated cells were then seeded onto the polymer scaffold and maintained in static bioreactors with conditioned growth media. Okay, now when these cells were allowed to grow, attached onto the scaffold, allowed to grow for around 2 to 3 days, and they underwent cell growth and differentiation. And once the cells have completely covered the scaffold in the duration of 2 to 3 days, they were taken out from the bioreactor in the lab, and the scaffold was implanted in the patient to act as a functional trachea. Okay, so now let's see an outcome of this study in this video. What you're going to look at over here in this video is basically surgeon's version of this whole study where they designed this trachea. Okay, so let's have a look at this video. Let's listen to this video, and then we'll come back and look at this specific case study, what happened in this case study, and what made this case to be famous all across the world in the field of tissue engineering.
With all previous treatment failing and no organ
Is the audio clear to everyone?
Yeah, we can hear the audio.
Science laboratory in Massachusetts, who had developed an artificial trachea. The patient's own stem cells would be used to coat the trachea, which would then act as a scaffold supporting tissue growth. Once the cells had been seeded, the trachea would be ready for transplantation. Harvard Bioscience President David Green.
We made something called the bioreactor, which is a box about the size of a shoebox, roughly this size, and that was where the trachea went from being a piece of plastic to being a piece of human tissue.
Scientists at University College London then became involved. Using 3D scans, they created a glass mould of Bayard's windpipe and his two main bronchial tubes. The mould was flown to Stockholm, where it was seeded with stem cells from Bayern's bone marrow.
The trachea, because it was seeded with stem cells taken from the own bodies, and there's no rejection, those stem cells live and they grow. And within a few weeks of the surgery, his own blood vessels have grown through the plastic scaffold as well. So it's really part of him today. It's his tissue, it's his cells, but those cells are differentiated from bone marrow cells and become all the different cell types that make up the trachea. So it really is a living, breathing organ at this point.
So as you could see in this video, so the patient was implanted with this artificial trachea that was grown in the lab. And as he just mentioned, they made use of stem cells and made use of other growth factors and differentiation factors to differentiate the stem cells into all the different kind of cells that are typically present in a trachea. And ultimately, these cells are growing on a scaffold that is shaped in the form of a trachea, right? So, and this at the end of the procedure, at the end of the process, the lab-grown trachea was implanted in the patient, and the patient survived the surgery. And it was actually claimed as a huge step forward for regenerative medicine because they were the first group of scientists who successfully executed the world's first synthetic organ transplant. And transplanting one organ from one patient to other patient is a much simpler process as compared to this case, because here you're not transplanting a functional organ from one patient to other patient. You're transplanting a synthetic organ that was completely grown in the lab, and then it was implanted into the patient, and then it was supposed to function the actual role which a typical trachea does in the humans. Okay, but let's see what happened after that. Okay, so as a result of this success, around nearly 18 surgeries were performed back to back within a span of 2 years from this time point. But within few months, what happened is a huge catastrophe happened. What happened is 8 of the 18 patients died. Whoever 18 patients who underwent the surgery, amongst them, 8 of the patients died because of severe complications, and the other 8 patients underwent alternate surgeries. They had to undergo alternate surgeries to remove the artificial trachea or come up with an alternative mode of survival. Okay, so very soon the study became famous as a failed clinical trial rather than being a successful clinical trial. And the publications which were associated with this work were retracted, citing manipulated results. And it featured for every wrong reason possible. Okay. Okay, but ultimately, let's see the reason for the failure of the graft and the loss of so many innocent lives in this whole case study. The investigation reported that the polymeric scaffold could not hold the sutures that was performed during the implantation procedure, and these sutures basically collapsed in many patients, which led to extensive damage to the tissues. Okay, so the detached tissues led to clogging of the bronchial passages through which we typically breathe, and ultimately led to the death of the patients. So in short, actually, if you look at this case, it is actually the mechanical incompliance of the polymer scaffold that was the reason for the failure of this trachea. Okay, so ultimately we come down to learn that scaffolds are vital components of tissue engineering, and thus it is important to understand the favorable aspects of the scaffold. For instance, in this case, it was the mechanical properties of the scaffold that played a pivotal role in leading to the failure of the implanted trachea, right? So what should be the rationale behind fabrication of a scaffold? That is what is presented over here. Now that we have seen different case studies of different organs, how they were transplanted, how they function, we've seen cases where there has been successful growth of a complete functional heart in the lab, as well as success in the growth of a complete functional bladder in the lab. But we have also seen the cases of failures, as in the case of the artificial trachea, which I just mentioned. Okay, so basically from all this study, we have to understand what should be the rationale behind fabrication of scaffold. What aspects should be taken into consideration when you're designing a typical scaffold? So what parameters have to be taken into consideration when we fabricate a scaffold? So this classification basically represents the complete list of such scaffold parameters. It is not just about what materials you use, it is also about how you fabricate it. What should be the surface characteristic of the scaffold? What should be the physical and mechanical properties of the mechanical as well as chemical properties of the scaffold? And we clearly saw that mechanical properties plays a very important role in determining success of any tissue engineer organ, right? And the scaffolds can be made up of, again, different materials. It can be metal, ceramic, synthetic polymers, and natural polymers, which depends upon the tissue or the organ that you want to grow. When the choice of these materials, it actually depends upon the target tissue that you want to grow in the lab. Okay, and these properties are all actually interlinked over here. Suppose you choose a biomaterial, it is expected to have better biocompatibility and biodegradability, at least better than metals and other composites. Likewise, when you choose between polymers and metals, you need to take into consideration the mechanical properties, because they are very different from these two materials. The mechanical properties are very different for these two materials. And the scaffold upon implantation should sustain the regular wear and tear that it experiences within our body, right? So let's look at the other aspects. Let's look at other aspects, whether they are biodegradable or not, and then at what rate should they degrade if they are biodegradable. If you talk about biodegradability of the scaffold, what it means is the scaffold which you have implanted in the patient, it has to decompose, and when decomposes, it should not lead to formation of any toxic byproducts, and it should be eliminated from the body. Okay, so which is what is a definition of biodegradable scaffold. Okay, so then once you design a biodegradable scaffold, you also have to ensure that it has to degrade at a rate that is proportional to the rate at which cells are growing within the scaffold. So if the scaffold degrades before the cells have grown, then the cells will not be able to take up the shape of a typical scaffold. So the rate of proliferation or migration or infusion of cells in the scaffold should be proportional to the rate of degradation of the scaffold. So it's not just that the scaffold should be biodegradable, the rate of biodegradation of the scaffold is also pretty crucial. Okay, so depending on how I want to tailor the degradation process, I would have to look at how I would like to cross-link it or modify the mechanical property, modify the degradation process, what type of material I can use so that the biodegradation process is altered to fit the tissue requirements. So there could be polymers, something like PCl, which have very slow degradation. You can use something like PEG, which actually just gets dissolved in a much faster way as compared to PCl. And depending on all these things, we can actually tailor the material to provide the desired properties. So these are all the properties which we think of from a biological standpoint. From the final production and manufacturing standpoint, we need a material to be processable, malleable, as well as it should also be sterilizable. What we have discussed so far is a typical favorable characteristics to design a scaffold so as to make it biodegradable, biocompatible. But when you design a scaffold, there are other aspects also that you have to look into, especially the production manufacturing standpoint of from the standpoint of production manufacturing, which means the material which you're going to use for making the scaffold should be easily processable. They should be malleable, and they should also be sterilizable. And so there are different techniques which you can use to sterilize the scaffold, but whatever technique you use to sterilize the scaffold should not affect the properties of the scaffold or the material which the scaffold is made up of. So if you're going to use protein on top of the scaffold, or for instance proteins like collagen, it should not get denatured when you are doing the sterilization procedure. Sterilization procedure is actually always performed for any scaffold that undergoes the transplantation into a living patient. Sterilization is basically getting rid of all the microorganisms and ensuring that the scaffold which you're going to implant into the patient doesn't have any infectious microorganisms present on it. For that, the scaffold is subject to different sterilization procedures, where either it can be subject to temperature or chemical agents and all those things. So when you do this temperature treatment or chemical treatment to sterilize the scaffold, you also have to ensure that the material that the scaffold is made up of doesn't undergo any mechanical variations. Okay, so and you can also functionalize these materials to get desired surface properties like hydrophobicity, and electrical properties, and so on. So all these things are key factors that determine success of scaffold for tissue engineering. So let's just touch upon one of these aspects, consideration of scaffold stiffness and how it governs the effects of cell characteristics. We'll just talk about only one aspect, that is how the scaffold stiffness impacts the morphology or the type of cells and how they grow and proliferate on the surface of these scaffolds. Okay, so the fate of cells is also affected by the stiffness of the surface microenvironment. To measure anything with precision, we need a highly sensitive instrument, just like that. In order to measure the effect of scaffold, we need a sensitive cell. And what could be more sensitive than a stem cells? Those of you who have first-hand experience handling stem cells would have realized that what I'm talking about. Okay, stem cells are pluripotent cells, meaning they can divide into more stem cells, or they can differentiate into any type of cells that is typically present in our body. So if you mess up with any minor step in culturing these stem cells, you will end up with totally different type of cells than what you intend to have. Okay, and this comic representation here represents the same scenario, where you want stem cells to differentiate into a neuron. You might end up with differentiating the stem cells into an Optimus Prime. Okay, with the slightest of changes in the culture conditions. So as we understand that the stem cells are more sensitive sensors for determining the effect of stiffness. So to understand how the cell proliferates or attaches and differentiates, you make use of stem cells and understand how the stiffness value determines the fate of the stem cells. So a lot of studies have been done in the past to show that the stiffness of the matrix could affect the differentiation spectrum of the stem cells. And that is what is shown over here in this figure. Okay, stem cells, if they are cultured in suspension, where there is no scaffold and thus minimal stiffness, they can differentiate into different blood cells, okay, B and T cells. With increasing stiffness of the scaffold, you can get whole new range of cells out of the same cells, same stem cells. Okay, so which can include the neurons, endothelial cells, fibroblast cells, muscle cells, chondrocytes, and osteoblasts. So from the same stem cells, you can generate multiple different kind of cells just by modulating the stiffness of the scaffold. Okay, so from this study, I think you can clearly understand that the mechanical property of the scaffold also plays an important role in governing the fate of the stem cells that you see on top of them. Okay, so now let's have a look at one or two one-on-one comparison in the published literature. In fact, in relation to this, there are articles showing that the same stem cell can differentiate into muscle cells on soft substrates and osteoblasts on harder substrates. Okay, so stem cells differentiate into muscle cells when the scaffold is made up of softer material. But same stem cells can differentiate into osteoblast, which are the cells that are present in your bones. And that happens when the scaffold is made up of harder materials. Okay, so there are another study that supported this finding, and stem cells on soft matter with a stiffness of less than, let's say, 0.05 kilopascal could promote the neural differentiation much more effectively, while hard stiffened materials or scaffolds promoted osteogenic differentiation effectively, which means formation of osteoblast and osteoclast cells. Okay. Again, people have illustrated the actual signaling process that happens in these studies. So this is what these are examples of different studies where they have shown that the matrix elasticity clearly determines what kind of cells you will end up with when you cultivate stem cells on these matrices. Okay, so a differentiation process of the stem cell is altered. The fate of the stem cells or the kind of differentiation that they will undergo will be different based on the stiffness of the scaffold that you have. And again, the migration of the stem cells is also governed by the kind of scaffold which you have. And of course, the third aspect of a typical cell is basically proliferation. The extent of proliferation is also controlled by the structure of the scaffold. Suppose our scaffold is porous in nature, which means the cells can proliferate and migrate through the pores, infiltrate into the scaffolds much better, unlike a scaffold which is not porous in nature. Okay, so all these aspects are very important. So in fact, as I was just mentioning, these articles basically summarize the importance of the scaffold stiffness alone in determining the fate of the stem cells that is growing on top of them. Okay, so likewise, every other aspect of the scaffold influences the cell's characteristics in its own and unique way, and a comprehensive note on each of these factors would actually go for multiple talks or multiple sessions. But with the given time limitation, I would like to limit the discussion here and add a short note of my inspiration figure in the field of tissue engineering, the person who defined the field of tissue engineering, Dr. Robert Langer. So just like a typical star of the day in cricket match, he can be called as basically an icon of the day. Dr. Robert Langer I think I mentioned about the same name, Robert Langer, in my first one or two slides, where we discussed about the vacanti mouse, right? Mouse with a human ear. So Robert Langer, as I already mentioned, he's a chemical engineer, and he's not only the pioneer of tissue engineering. He's pioneer of tissue engineering for two facts. First fact is obviously he was the first person to contribute to the development of vacanti mouse, and apart from that experiment, he's also known for the highest number of citations ever achieved by an engineer so far. So if you look at the citations and number of articles that he has uh, published so far, he is a person with the maximum number of citations at present in this whole world. So such is the impact of the scientist Robert Langer, and such is the impact of tissue engineering also. So you take any discipline, he is the person with maximum citation, and it not just justifies his contribution, it also justifies the contribution of tissue engineering in our recent research and development aspects. Okay, so and then he certainly speaks about his contribution. Uh, he also has, as I said, he has a maximum citation, and he speaks about his contribution to the field of tissue engineering at many avenues and many platforms. And apart from this citation aspect, if you look at other staggering figures associated with him, he has nearly 1,260 patents in the field of tissue engineering, and he's also founder of nearly 40 startups. And all these 4 startups have come up from his lab. And I had basically a dream come true moment when I had an opportunity to attend his lecture at Stanford. This was during 2018 November. It was nevertheless an inspiring lecture, but I also had an opportunity to ask him one thing, which which I'd like to pursue in my career also. Uh, I asked him how he manages in being involved in so many companies, as well as how he manages to manage the huge research group also at the same time. So let's see what he has to say. And with this video, we'll try to conclude a lecture. So this video is basically a recording of the same session where I attended his lecture. So you'll see me asking a question, and then he'll be responding to that question with a simple answer that give your research scholars some ideas, turn them loose, and try to be there when they need your advice or help. That's all he does to manifest such a great outcome in terms of research. So many publications, so many startups, all those things, just by this simple principle. So I'll just play this video. At the end of this video, we'll basically conclude this lecture. So, okay. So video is not playing. I guess there's some issue with my player. But anyway, as I said, the answer to my question that how he manages such a huge research group as well as so many startups at the same time was that he basically gives new ideas to his researchers and then lets them loose, which means he basically gives them freedom to try whatever they want. And then he tries to be there when they need his help. And with all just these minimal things, he's able to manifest such a huge input or impact on the tissue engineering research. Okay, so with that high note, I would like to conclude today's lecture. So, and I would like to open the session for further questions and discussions.
Thank you, Professor. It was a very wonderful presentation. It was really awesome. You have detailed everything from the scratch, and you have inspired us to have a look on the scaffold design for in the tissue engineering. I hope participants would have enriched their knowledge based on your input. And participants, we have questions now. Kindly put your questions in the chat, or you may unmute yourself and ask your questions.
Sir, good evening, sir. Very nice presentation, sir. I'm having one doubt, sir. Basically, for our research, my student PhD scholar is doing electrospinning techniques for hydrogel preparation. along with the
Nice.
nanoparticles, sir, for wound healing.
Okay. Correct.
So
Okay.
whether that will be used for as a scaffold, or do I need to purchase a already existing scaffold and then coat my nanoparticles or plant extracts for wound healing, sir? What you suggest, sir?
So electrospinning is a very interesting technique. In fact, I worked on this technique during my PhD also. So this technique is basically used for fabrication of nanofibers, which I think you have already mentioned, and it's a very the nanofibers are a very appropriate kind of structural material that can be used for tissue engineering. And the reason for that is these fibers, which are in nanoscale dimensions, they exactly mimic the structural nature of the extracellular matrix. As I said earlier in this lecture, the extracellular matrix is made up of structural proteins, collagen, fibronectin, and all these things. But these structural proteins are again present in the form of fibers, and they form like a mesh on which the cells are present or attached. And these nanofibers mimic the similar mechanical property, similar physical property as that of an collagen or fibrinogen that is present in your actual tissue. So for the structural feature or structural aspect of the nanofibers, with respect to that, they form a very good scaffold. They act as a very good scaffold for tissue engineering purposes. So, and if you're talking about loading bioactive agents into this scaffold, into these polymeric nanofibers and making use of it for different applications, there are different ways people have done this. Some of the people, what they typically do is they add the bioactive agent into the polymer solution, and then draw nanofibers from this polymer solution. And then when you have such a nanofiber, you will have all your bioactive components spread throughout the nanofiber that is being formed by electrospinning process, right? So that is one way. Other way is form the nanofiber and then add the bioactive agents on surface of these nanofibers. But then in that case, you have to adsorb the bioactive agents on the polymer nanofibers. For that, you have to have a strategies just to ensure that the release profile is consistent with the requirements of the application. So these two possibilities are there, but it also depends upon what kind of release profile you want. You can also cross-link the polymeric fibers so that the release profile is slower or faster according to your need. Yeah, so maybe there are multiple strategies to incorporate bioactive agents in the nanofibers. Yeah, I hope I answered your question, or
Yes, sir. Thank you, sir.
okay, thanks. Is there any other questions which I can answer, or?
Participants, if you have any question, you may ask. We will have another 2 minutes for the question session. And Professor, if we want to work on the scaffold preparation, yeah, is it require any advanced or sophisticated materials to be in the laboratory, or can in an academic laboratory we can try on the scaffold preparation and characterization aspects?
Yeah, scaffolds. As I said, there are different. We can make an artificial scaffold, which is basically a synthetic scaffold, or a scaffold that is derived from biological materials as such. So, if you're talking about scaffolds made from biological materials, you can extract the proteins from different biological sources and then make a scaffold from these proteins or carbohydrates and all those things. And apart from that, the other possible way would be to make use of synthetic polymers. Like, as I said, there are some polymers which are FDA approved polymers, which means they are biodegradable, biocompatible, and we can process these polymers to take the shape of different scaffold features, and then use it as a template to see the cells on top of them and then grow further. But again, if you are talking about growing the cells, again, there are specific requirements in terms of biosafety and all. So BSL 2 level safety has to be adopted, and while handling cells, especially mammalian cells and all those things. And then, but if you're talking about animal implantation of these scaffolds, that requires another bigger round of regulations and all those things. So animal trial approvals are all is required if you want to test your scaffold in animals and all. So such regulations are involved. So usually what people do is we don't have not many institutes have animal facility. So they design the scaffold, do some cell culture study in their biosafety level 2 lab facility. And then once they find promising results over there, they send the samples to other institutes where they have animal facility and all. They can do the testing animal study, and then people interpret whether it can be translated into clinic and all or not. Yeah.
Yeah, understand. Thank you. Thank you, Professor.
Okay. Yeah. Thank you.
And we have another question in the chat, Professor. If you could
Okay.
see.
If you want to prepare nanoparticle infused hydrogel for wound healing, what techniques do we need to do apart from antimicrobial activity? Infuse a wound healing. Okay, so I hope I understand the question properly. So basically, you're trying to make use of hydrogels for hydrogels with some antimicrobial compounds, especially for wound healing applications. So having an antimicrobial property for a scaffold for wound healing is reasonable because obviously you don't want the bacteria to grow there in the wound. So that you're able to achieve by adding antimicrobial compound. So I guess your question is basically what other properties can be included in such a scaffold that is used for wound healing purposes. So maybe the other aspect which you can look at is whether your scaffold is able to give an idea about the progress in the wound healing procedure. For instance, all of us used a Band-Aid to close a wound, right? But we close a wound with a Band-Aid, but we are not able to see whether the wound is healing or not. Okay, if you want to see whether the wound is healing or not, you have to remove the Band-Aid and then have a look at it. So what you can do is when designing such a scaffold for wound healing, you can add some components or additional nanoparticles which are sensitive to pH modulation. So when the wound is undergoing a healing process, the pH trend is different. But when the same wound is undergoing infection process, the pH trend is different. So when you incorporate such a pH sensitive nanoparticle in your wound healing scaffold and you place it on top of your wound, so if the wound underneath is undergoing healing process, because of the nanoparticle which you have added over there, because it is pH sensitive, it will exhibit a different kind of color. And if the wound is not undergoing healing process, it is infected, the pH would be different. And because the pH is different now, it is expected to show a different color. So basically, you can incorporate a sensor on these wound healing scaffolds, which gives you an idea whether the wound underneath is undergoing healing process or it is getting infected. Okay, so because we don't have such a scaffold in the market, which helps us in understanding whether the wound underneath is healing or it is getting infected without removing the Band-Aid or whatsoever it is. Yeah.
Great. Thank you, Professor.
Okay. Thank you, everyone. I hope the session was informative. Yeah.
I take this opportunity to thank Professor Uday Kumar for sharing his valuable content here. And thank you, Professor. Yeah.
Thank you.
Participants, we are winding up the session today. We'll be meeting you by tomorrow, 6 p.m. sharply. Thank you for your patience and valuable time. Take care. Thank you, Professor.
Thank you.